Hi, hello, I'm Shishra Joy, second MSc Biology student, St. Mary's College, Thrissur. Now we are discussing about the microbial locomotion. Bacterial movement, gliding motility, amoeboid movement. These are the microbial locomotion. So, first we discussing about the bacterial movement. Most of the bacteria are motile with help of flagella. It is ATP dependent process because it needs energy. To identify motile bacteria, the pattern of flagellation is very much important. Prokaryotic flagellae much thinner than eukaryotic flagellae. It contains 78 to 81% protein, 13 to 23% lipid, 1 to 6% carbohydrate. So the structure of the flagella. It is pipe-like structure. Three type of flagellae used for locomotion. They are structurally different. 20 to 30 genes involving for the synthesis of flagellae. So it has main three parts. This is the outermost region of the flagellae. It is the filament. The next part is the hook. Hook attach the filament. Then third part is the basal body or motor device. It is anchor of flagellum to the cell wall and plasma membrane. So it has mainly three parts: filament, hook, basal body. Filament, it is long outermost region of the flagella. It is composed of the protein called flagellin. It is arranged seven intervened chains that form a helix around the hollow core. Then next one is the hook. Filament is attached to the hook. It consists of different type of proteins. Then next one is the basal body. It is anchor the flagellum to the cell wall and plasma membrane. It is composed of small central rod inserted into a series of rings. Gram-negative bacteria have two pair of rings and gram-positive bacteria have one pair of rings. So the mechanism of the bacterial movement. The filament is a shape of a rigid helix and the bacteria move when this helix rotates. When a screw is turned, it moves forward like the filament in the bacteria helically rotate. The filament is helically rotate. The bacterium is moved forward. In this manner, locomotion is possible. Flagellar rotation determines the nature of bacterial movement. Then next is gliding motility. Flagella independent motility. There is no need of flagella. Slower and smoother than swimming. Movement typically occurs along the axis of the cell, requires surface contact. Specific motility membrane protein help for the gliding motility. Some proteins in cytoplasmic and outer membrane help for the motility. The next one is the pili. So what happened to the pili? Pili attached to the cell as well as the surface. It's need a surface. Pili attached to the surface. When it is in the stable condition, the proteins in the pili broken down. So what happened? The pili shrinks. So this way the cell closer to the surface. So in this way the locomotion is possible. The pili pulling the cell closer to the surface. The next is the polysaccharide jet. The polysaccharide slime is secreted outside the surface of the cell. So this is the polysaccharide slime secreted by the cell. It attaches to the cell as well as the solid surface. When it is in the stable condition, the slime adheres. The cell is pulled along the surface. Then next is the amoeboid movement. Amoeboid movement is the movement of an entire cell in the relation to its surroundings. It's received the name from the fact that amoeba move in this manner. So mechanism of the amoeboidal movement. So there is a protrusion of pseudopodium from one end of the cell. In one end of the cell there is the protrusion. So partial content of the cell is moved forward to the protrusion of the pseudopodium. So there is attachment happen, there is a stability occurs. What happened? The entire cell content is moved towards the pseudopodium, the protrusion of the pseudopodium. The membrane of the end of the cell is continuously moving forward and left end of the cell is continuously following along as the cell moves. So, two effects are essential for forward movement of the cell. As we said about the attachment of the pseudopodium to surrounding tissue, so that it become fixed in its leading position, while the remainder of the cell body is pulled forward the point of attachment. The second essential effect for locomotion is provide the energy required to pull the whole cell body in the direction of the pseudopodium. Example, white blood cells, fibroblast. Then taxes, directed movement in response to the chemical or physical gradient. Chemotaxis, a response to the chemical. Organism is moved towards the chemical. The phototaxis is a response to the light. Organism move towards the light. Example, trees grow towards the light. Then aerotaxis, a response to the oxygen. Osmotaxis, a response to the ionic strength. Hydrotaxis, a response to the water. Thank you.